Now it's my honor to introduce Dr. Saki Takahashi. Saki Takahashi is an assistant professor in the Department of Epi at the Johns Hopkins Bloomberg School of Public Health. She is broadly interested in developing mathematical and statistical models closely linked to empirical data to understand how immunity shapes the risk, burden, and spread of infectious diseases, and to harness these insights to inform public health interventions. Dr. Takahashi's talk today is entitled Integrating Insights from Multiplexed Pathogen Surveillance into Epidemiological Models. Take it away. Thank you.
Thanks so much. Thank you so much for the introduction and the invitation to present. I'm absolutely delighted to be here. I actually first got my start in infectious disease epi and modeling as an undergrad here a number of years ago now, as a summer research intern at CCDD with Mark Lipsitch and Ed Goldstein. So it's really, really nice to be back. And yes, so now I'm an assistant professor at Hopkins. I've just started there this past fall and eagerly recruiting students and postdocs as well. So just wanted to broadly highlight the types of questions that I'm interested in my research. And so really, I kind of think of it as three different buckets. First one being thinking about population level susceptibility and transmission. The second kind of scale being more at the individual level, thinking about how different pathogen exposure histories and really thinking about like how those two scales link. So individual informing population and vice versa. And then, of course, the ultimate goal is to quantify disease burden and inform different control interventions. And so in my work, the tools that I use broadly are public health surveillance data. And increasingly, I'm also involved in various field epidemiologic studies, particularly cohort studies, to try to answer some of the questions. And then I use various modeling tools. And in this talk here, I'll also highlight some of my ongoing work using different types of pathogen detection tools as kind of an additional piece to try to answer these questions. And I will emphasize some of these kind of multiplex tools that we are using increasingly. And so I think everybody here is very familiar with this, but I just wanted to get us on the same page when I talk about different approaches to pathogen detection. And so first, obviously there's direct detection, which is diagnostic antigen molecular testing for the presence or absence of pathogen. And there are a variety of ways, right, that we do this, whether it be, so one of the pathogens I work on is malaria, where we do microscopy for detection. But of course, the other tools being things like qPCR or amplicon sequencing. And in this talk, I'll highlight some of the work that we've been doing using metagenomic next generation sequencing or mNGS to do molecular pan-pathogen detection. So just to highlight, so this technology, mNGS, has been used previously for a number of different use cases. For public health, it's been used for identifying different novel pathogens or identifying things like co-infections. There are also examples of using mNGS to detect outbreaks. And then more broadly, as a research tool, it can be used to characterize the microbial landscape. So I'll talk more about the assay in a bit, but broadly, it can identify different types of microbes from viruses, bacteria, eukaryota. And so this here on the left is a fairly recent analysis investigating pathogens detected in pediatric febrile illness cases in an inpatient setting in Uganda. And so this was using mNGS to try to characterize those different potential causative agents. And then another tool that I use quite frequently is indirect detection. So this is also, this is particularly serological testing. And so in serology, we test for antibodies, which provide some record of an individual's pathogen exposure history. And so tools here include everything from ELISAs to bead-based assays to much more multiplexed assays such as protein microarrays and phage arrays. So this is just a schematic. So a lot of the work I do involves the use of cohort studies. And so this is just a cartoon showing 3 individuals, right, from a cohort. And let's just imagine we do routine serum sampling, let's say every 3 months or so. And then the presence of one of these red antibodies, right, indicates an individual having antibody responses to that red pathogen. So here we just have 3 individuals, right? And so if you're testing for antibodies, that's the indirect approach. And then let's say here for this 3rd person, we also observed an infection event. So let's say they had a febrile episode, we detected a pathogen. And so now, right, so we detected that pathogen, and let's say that those antibody responses are to that particular pathogen. And so this is like very, very simple, like simply what can we do with these different types of data? So one approach that we take fairly often in very applied settings, right, is to estimate seroprevalence. So what proportion of that population has detectable antibodies to that pathogen? Another one being we can do things like try to estimate the attack rate, right? If we have 2 different time points, we can try to estimate what proportion of the population has seroconverted or has seen that antigen before. We can also do things like estimate case ascertainment. So what proportion of infections are we detecting using direct approaches? And so this is not at all a new idea, but something that I'm quite excited about. So trying to link the different types of detection, so direct detection with serology, and particularly within the setting of longitudinal studies. So obviously, right, direct pathogen detection can tell us about things like viral load or pathogen load, parasite densities, what pathogens individuals are infected with and when, and all those sorts of things. And then with serology, we can answer complementary questions about cumulative incidence or course of infection. And then in terms of trying to link the two, I think this is a fairly often used analogy, right, of this tip of the iceberg. I think serology really can fill in some of the gaps on these partially observed processes. And then the other way around, so this is an analogy. So I used to play violin when I was a kid. And so I think about kind of how direct pathogen detection within a cohort could be used to generate gold standard samples for serology by providing time since infection. So really the analogy here is quite silly, but like the idea is to kind of tune your instrument to the right frequency so that you anchor the rest of the pitches. And so that's really what direct pathogen detection is doing within a cohort, is kind of anchoring samples to how recently an individual has been infected or exposed. And so just another way of saying that, right, is to just to show here, right, if we have an observed infection or detected infection, then the time points before and after become very valuable in terms of calibrating how recent or the time since or prior to infection. So this can help us inform models of antibody kinetics. So in today's talk, I will present 3 different vignettes, kind of trying to link these ideas together. The first one is a study that we recently did, trying to investigate causes of non-malarial fevers in a high malaria burden setting using metagenomic sequencing. I'll then talk about some of our COVID serology work. So in particular, I'll present some of our methods trying to estimate cumulative incidence for SARS-CoV-2 from population serosurveys, accounting for waning of antibody responses. And then if I have time at the end, I'll present some work where we've been trying to link direct pathogen detection with serology to try to reconstruct infection histories. So for the first bit. All right, so So in this project, we've been thinking about how to characterize etiologies of non-malarial fever in a high malaria burden setting. So I think, so as you may know, fever is a very frequent disease manifestation, particularly in children. And so fever has many different underlying etiologies, right? Viral, bacterial, eukaryotic. And and one of the challenges of fever is that it's a non-specific disease presentation. So we need diagnostic testing if we want to understand to quantitate the different causes and understand the etiologies. And in the setting of, or in high malaria burden settings, this is complicated, right? Because malaria obviously is a major cause of morbidity and mortality. And fever is also a very common symptom of uncomplicated infection with malaria. Luckily for malaria, recent increases in diagnostic testing, so both microscopy and molecular methods, have led to improvements in malaria case management. But the challenging thing is for fevers that are tested and don't result in a malaria diagnosis, there are these huge knowledge gaps in terms of the etiologies, and this is because of limited availability of testing for other pathogens. And so this limited availability of testing leads to a number of different things. It leads to undiagnosed illnesses, missed opportunities for targeted treatment, unnecessary empiric use of antibiotics, and also public health surveillance systems that provide an incomplete picture of the pathogen landscape. There's also an additional challenge to studying fever in high malaria burden settings, which is there's individuals who are exposed often to malaria will develop clinical immunity against malaria, which means that those individuals will be able to tolerate the parasites in their blood without developing fever. And so in high malaria burden settings, individuals who have parasitemia for malaria, but who have acquired high levels of clinical immunity, they're unlikely to have malaria be the cause of febrile illnesses. And so, like I said, I work on a number of cohort studies. So this study here was conducted within the PRISM border cohort study in Eastern Uganda. So this is a high malaria transmission setting. And this cohort study, which began in August 2020 and will carry on until September of this year, is a representative longitudinal cohort of 80 households, around 500 participants who are enrolled into this cohort. And it's a malaria surveillance study. And so there's both active and passive surveillance for malaria, active monthly surveillance, as well as continuous passive surveillance. And so individuals are present at the dedicated study clinic every month as part of the study. And then they're also encouraged to attend the study clinic for all their healthcare needs, which means that ideally we are capturing a lot of other infections that they might be experiencing as well. And one of the amazing things about being embedded in a study like this is we have extensive clinical data, both on individual symptoms, as well as other information like vaccination records. And there's also a very rich biospecimen repository on which we can ask additional questions. And so in this study, where we were looking at causes of non-malarial fever within this cohort, what we did was we designed a study where we collected respiratory swabs and plasma, so paired samples collected at all visits where individuals presented with non-malarial fever. And then those samples were tested using metagenomic sequencing at the Chan Zuckerberg BioHub. And so all of the laboratory work and a lot of the analysis was done by a research associate, Asaj, who's shown up here, who did a lot of amazing work for this study. And then I'll quickly present the mNGS pipeline, like happy to discuss details later, if folks are interested, but broadly, right, this is just showing the pipeline for metagenomics, starting from study design, where we preserve the samples in RNA DNA shield, And then, so the starting material is DNA or RNA from a sample, and then library prep, and then all of the genetic material is sequenced and then analyzed using a bioinformatics pipeline called IDSeq, now known as CZID, that does a lot of the downstream like QC, host removal, ranking of hits, whatnot. And so ultimately the outcome is to identify the different microbes that are present in each sample. And so one of the findings, even prior to mNGS, is that the majority of febrile episodes were negative for malaria parasites by microscopy. So as you can see, the bit that I'm presenting is just on the first 8 months of this study. So December 2020 to August 2021. As you can see, we had thousands of study visits conducted during this time period. And 624 of those visits, individuals presented with fever. Over half of the time, so 57% of the time, those visits with fever were blood smear negative for malaria by microscopy. And so those individuals who presented at those visits were enrolled into our mNGS study. And then we obtained plasma and swabs from those individuals. And then, so the rest of the time, 43% of the time, those individuals tested positive for malaria by microscopy. And so they were not enrolled into this study. Or into our metagenomics study. And so, so the output of these assays, just showing here on the left, so this is jumping right into the results, but these are the human viral species that we identified. I have another figure here where you'll be able to see those a little bit more easily, but just to kind of orient folks, the output of these assays is going to be about like the frequency of species identification, but also you get quantitative outputs like the normalized NT read count. You can also get the breadth of the genome recovered, and if you have enough reads, then oftentimes we are able to recover genomes, particularly for viruses. And then you might be wondering how these read counts correlate to something more gold standard like CT values. This is not an analysis that we did, but this was a study that was done to compare the outputs of mNGS to gold standard PCR CT values. And this was for SARS-CoV-2 in particular, where they found that essentially any CT value that was higher than 32, this mNGS assay does not seem to catch well. But if it's lower than that, then it does seem to be quite sensitive. And so I'll just say, I think these kinds of validations are going to be increasingly important, right? If we want to be able to to really interpret the results of these assays. So just jumping into the main findings, we identified co-circulation of SARS-CoV-2 and multiple respiratory viruses during this time period. You can see here, this is just a histogram of the different pathogens that we identified, including rhinoviruses, which were the most prevalent at 40 detections, also flu A, paraflu, RSV, SARS-CoV-2, and 3 of the 4 seasonal strains of seasonal coronaviruses. And it turns out that during this time, which was during the Delta wave of SARS-CoV-2 in so May to July 2021, we identified actually more flu cases than we did SARS-CoV-2. And so the really cool thing, or one of the cool things we can do, is like generate consensus genomes. And so we did some phylo analysis of the genomes that we were able to generate as well, and found that the viral diversity did kind of cluster with other sequences from the region, as you might expect. And so in terms of what we were able to detect, or at how often we were able to detect viral pathogens at these visits, overall, around 39% of visits, we were able to detect a viral pathogen during a non-malarial fever. And that kind of scaled with age. So in kids under 5, almost half of those febrile episodes, we were able to identify a viral pathogen, and that kind of decreased with age. And so here it's just showing the kind of the composition of the different viruses identified by different age groups, so under 5, 5 to 15, and 16 plus. And so just to give a sense of how this compares to public health surveillance data, the top figure here is just showing counts of COVID-19 reported by the MOH, and then the bottom is counts of flu A, particularly the H3 subtype, from the WHO flu surveillance system. And of course, it's it's hard to directly compare these data sets, but just by eye, right? You would imagine that they would vary by orders of magnitude. But in this kind of very systematic and comprehensive study that we did within this small cohort, and we had small sample sizes, but that kind of suggests the relative numbers of cases might be more similar. And so overall, in terms of conclusions for this piece, we think that there's utility and potential of doing multiplex pathogen detection as a tool for surveillance, as well as for better understanding the overall burden of different pathogens. I think a key limitation of this work was our inability to look at the contribution of bacteria and how bacterial microbes could be contributing to non-malarial fever. We are starting to address this now from a study design perspective, where we are collecting convalescent samples from individuals to try to establish what the background commensal microbiome model might look like, to be able to try to tease apart pathogenic from commensal bacterial microbes. This is still very much a research tool, so we'll need to there will need to be continued validation against gold standard metrics, and additional testing is still ongoing as we have a couple more years of sample collection. All right, so I'm gonna switch gears a bit and now focus on some of the work that we've been doing on serology, particularly in the context of SARS-CoV-2. So I think folks here again are quite familiar with this idea, but serology is the gold standard to try to quantify infections in a population. Antibodies are used as a biomarker of past exposure, right? And they can measure who's been infected or vaccinated. And if you have a correlate of immunity, they can also measure who is protected. So this is often done with things like VPD, vaccine preventable disease. So antibodies allow us to estimate various transmission parameters and to understand the natural history of pathogens. So things like the case ascertainment ratio or infection fatality, as well as the epidemiology in terms of seroprevalence and downstream metrics like FOI, course of infection. But all of this is kind of underpinned by having the right assay to answer your question and the ability to adequately interpret the results of your serological analysis. And so this is just to highlight that during the SARS-CoV-2 pandemic, serosurveillance has really taken off around the world. I mean, serosurveillance was done prior to COVID, but I think that if you just look at some of the numbers of serosurveys that have been conducted since the pandemic began, it'll give you a sense of, there are thousands of serosurveys that have been done for SARS-CoV-2 in both at the kind of domestic scale as well as the global scale. These are some of the two resources that aggregate that information if you want to take a further look. But one of the things that when I was a postdoc at UCSF, we spent a lot of time thinking about is if seroprevalence estimates for SARS-CoV-2 could be biased, and this is the thinking. And so in the general population, which is where we often want to have our serosurveys be representative to, there's a spectrum of disease severity. So we expect those individuals to have had varying magnitudes of antibody responses. So individuals who had more severe SARS-CoV-2 likely have had higher antibody response than individuals who had mild or asymptomatic infection. And then there's also this issue of time, right, where antibody responses typically wane over time since infection. But something that we and many others noticed early on in the pandemic is that the positive controls that were being used for validation were limited to early convalescent samples from patients with severe disease. So those are typically going to be individuals who have the highest antibody responses. And so we did some work at the beginning of the pandemic and to kind of so we've been trying to think about how we need to account for this reduced sensitivity of different serological assays in the general population in order to avoid this idea of spectrum bias. And so one of the things that we've been doing is developing different methods to do estimation of cumulative incidence from serosurveys, incorporating these this idea of thinking about how antibody kinetics wane as a time since infection, and then thinking about the recency of the epidemic in a population. And so it's a very, very simple framework, but essentially, if the goal, which is shown on the right, is to accurately estimate cumulative incidence, and this is from a serosurvey with binary serostatus data, so just ones and zeros. And so if that's the goal, then what we need to do is come up with a weighted assay sensitivity that represents the sensitivity of that assay in that population at that time. And then the two kind of pieces that inform that are assay performance, so thinking about how sensitivity changes as a function of disease severity and time since infection, and then thinking about the local context of the serosurvey. So kind of the date, the place, the assay that was used, as well as the epidemic trajectory in that place. So just for this first bit here, thinking about assay performance and how that contributes to sensitivity. So longitudinal post-infection cohorts are really the gold standard to be able to try to quantify what kinetics looks like. So this I'm just showing from a post-infection COVID cohort out of UCSF that I was involved in. And one of the assays that was used was this ARCHITECT assay that was looking at IgG responses to the nucleocapsid protein to SARS-CoV-2. And so on the left here is just is the data from the longitudinal cohort. So looking at antibody responses as a function of days since symptom onset, stratified by severity, or here using hospitalization status as a proxy for severity. And you can see quite clearly individuals who had more severe infection also had the higher, antibodies responses shown on the Y, and hospitalized individuals had kind of the highest antibody responses, and in the non-hospitalized population, that was you have both high responders as well as low responders. And the purple line here is showing the cutoff for seropositivity established by the manufacturer. And so you can see that there's quite rapid waning, and you can imagine how the sensitivity of this assay, right, might be quite high at the beginning, but then would decline quite a bit as time since infection or time since symptom onset. And so we kind of threw this into a Bayesian hierarchical mixture model shown by these equations here, where we essentially assumed that there were high responders, low responders, and then tried to estimate sensitivity by both severity and time since infection. So I'm happy to discuss the model later if folks are interested, but essentially what we can do is estimate sensitivity, kind of incorporating these various sources of uncertainty, and come up with sensitivity curves as a function of time since symptom onset, as well as severity. And so it's just worth noting that the actual reported sensitivity of this assay was 100%, but you can see here that the sensitivity that we estimate from the post-infection cohort estimates that sensitivity will be quite low, a year post-infection. And so we need to think about these things, and this assay was used quite often for some of the biggest serosurveys, especially at the beginning of the pandemic. So it's just worth thinking about, right, how the sensitivity that is reported versus the sensitivity that might exist, or the actual sensitivity if you were to use that for a serosurvey might be quite different. So this last bit here, for the second contribution, is thinking about the serosurvey itself. And so you can expect, right, So here I'll just show, we have to think about the recency of infection in the population that the serosurvey is being done in relative to when the actual survey was done. And so now this is like quite old data now, but this is just looking at the states in New England here, looking at daily reported deaths due to SARS-CoV-2. And then I've just shown here the first 4 time points of the CDC commercial lab serosurveys that were done. And so you can see, right, the serosurveys were done or were started quite after the peak, the first peak in spring 2020. And so you can imagine just taking the previous sensitivity curves and kind of thinking about how these serosurveys, right, happened quite a bit after the peak. And so you can imagine that would affect sensitivity. So I won't go through all this, but we also, we looked at daily reported deaths as kind of a stable metric, at least during this time period of the epidemic trajectory. Clearly, there would be a lot of issues with applying this method directly now. But at the time, so essentially we tried to take the death curve and then recalibrate that, or to shift that over to be the symptom onset curve so that we could relate it to our serology data. And so effectively what we did is infer the counts of symptom onsets at different times. And so we can put that together and essentially come up with a single weighted assay sensitivity. And so what this method does is it'll create an adjusted or a weighted assay sensitivity that incorporates both how recently people in the population were infected, as well as the sensitivity, and then like come up with a new sensitivity that you can use to adjust your serosurvey estimates. And so we applied this method to various sources of serosurveys that we knew used this assay, the one that wanes quite a bit. And so we can do things like calculate the ratio of cumulative incidence to seroprevalence. In this particular case in New England, we saw that it was around a 2-fold difference between raw and adjusted. And then we also did different simulations to try to quantify the effects of both how long it's been since, or what the trajectory of the epidemic looks like, and the proportion of the time that you use this assay compared to something that might be more robust. And so the heat map here is just showing the weighted estimates of sensitivity. We, just taking these model outputs, you get sensitivity to be quite low. But it's also worth mentioning that CDC did switch to using other assays that are more robust over time, sometime in 2021. But you can see here, right, that sensitivity would be quite a bit lower than expected with continued use of these kind of with this particular assay. So just to summarize this bit here, so interpretation of serosurvey results requires thinking about both the population level trajectory of the epidemic as well as individual level antibody kinetics. And this approach has also a number of limitations, one of them being assuming temporal homogeneity in case fatality ratio. Like I said earlier, I think there would definitely be issues with doing that now. A lot of things have changed as well, right? Where now the distribution of antibody responses in the population are going to be a lot more heterogeneous due to vaccination, multiple reinfections, boostings, et cetera. But I think something that does kind of underpin this is that that post-infection cohorts are going to be key and have been and are going to be key to be able to characterize longitudinal performance. Right.
I know you have another section.
Yeah, sorry, I'm going a little over. So, okay, so I will just end so here's the summary. Of course, I just wanted to acknowledge all the folks who were involved in this work. A lot of this was done when I was postdoc at UCSF, as well as now I'm at Hopkins, number of folks there to thank, and then funding sources. Thank you so much.
Awesome. Thank you very much, Dr. Takahashi, for a really great talk. Yeah, so we have some time for questions right now. As a reminder for the people on Zoom, send your questions to Kaya Joshi. Let her know if you want to ask the question yourself. But we can start with some questions in the room. Yeah, you want to talk?
Thank you for the interesting talk and great work. I'm curious for the first project in the metagenomic analysis. Did you know that you focused on the people who had fever and not malaria? But I wonder, did you look at anyone who also had malaria or people who are not febrile, just see what the carriage rates for some of those viral infections were, asymptomatic, perhaps?
It's a great question. Thanks. Yeah. So for the first, so for the population of individuals with malaria in detected by microscopy, we didn't. I mean, I think that that's also another limitation that prevents us from saying that this is the incidence of these viruses, because we didn't test them. We are now looking at samples collected from individuals after their non-malarial fever. So after the fever has, after they're no longer febrile, so about 6 weeks post-infection, to get at this question of carriage and also viral infections in individuals without fever, since I think we expect for rhinoviruses and some of these, rhinoviruses in particular, to not necessarily be the cause of the fever. So yeah, but I think we're hoping that by testing acute and convalescent samples, we will start to be able to look at carriage and maybe also say something about bacterial pathogens as well. The other kind of cool thing that we're thinking about now is potentially looking at the host transcriptome to try to tease apart viral versus bacterial infections. At least we have the gold standard or some gold standard viral infections. So maybe if I don't know if we will be able to do it, but if we can kind of tease apart signatures of a viral versus non-viral infection, then that might also give us some more information.
We have a question
Sure.
from the chat. So this question is from Caroline. For those with non-malarial viral fevers, does your clinical data include information on antibiotic prescription to potentially assess overprescription? Do you have any thoughts on how rare co-infections of viruses and antibiotics would be?
Thanks. That's also a great question. In the cohort, we do collect information on antibiotics that are prescribed. So we're just starting to look at that data as well, because of course that's also going to that's going to be a key piece of trying to say anything about the bacterial microbes that we detect. So yes, so definitely we're planning to look into that. And then in terms of viral co-infections, so we did detect some instances. They were not necessarily that surprising. It's often like a rhinovirus with something else. And again, like rhinoviruses, I think they are quite prevalent. And so I don't know if it's we're just detecting a rhinovirus and the cause of the fever is something else. If it's like SARS-CoV-2 or influenza or something like that, that we might be more confidently be able to say that it's kind of causing the fever. I will say also that since we were only looking at fevers, we do have other clinical information as well. So we do have information on respiratory symptoms and whatnot. And so I think fever is just like the way that we use to design the study. But I think if we wanted to look at respiratory infection or GI symptoms or something like that, then that would also be like some really interesting extensions. And we could probably look for other pathogens as well.
We have time for maybe one more question.
No one else. For the second project, I was curious about the time period over which you were doing this serological analyses and the relationship between the serological assay and the circulating variant. Do you know if the titers change as the variant, perhaps the ratio of the spike changes as well? And if I think it was spike that was the basis for the antibody detection. So yeah, curious what's the relationship is between the assay, the circulating variant, and your estimates of seroprevalence.
Thanks. Yeah, so the work that I showed was only done during kind of the first or the second half of 2020. So it's all kind of wild type. I think that, yeah, it would be really interesting to be able to look at kind of variant specific effects in a project that I did not have time to share today, where we did some longitudinal sampling within the same cohort study in Uganda for SARS-CoV-2. We were able to use a spike assay, so looking at wild type spike responses in kind of like in the first from 2020 to early 2022 in the same cohort. And so, yeah, there's some, I think, I guess I'll just say that, yeah, I think variant specific assays are probably are needed, right, to be able to really kind of to pin down, yeah, I guess, immune landscapes in a way. But yeah, I think that in this particular case, we were just looking at wild type, so not really kind of getting into those intricacies.
All right, we're going to sneak in one more question.
Okay, so this is from Nancy Leon. She says, thanks for your talk. And her question is, for estimating SARS-CoV-2 seroprevalence, how do you account for vaccination history? And in parentheses, highly seropositive between consecutive blood draws.
Yeah. So I guess it depends. Very much on the question that you want to answer. So again, I didn't get a chance to talk about this, but we did do some work trying to estimate attack rates of SARS-CoV-2 in Uganda. And so there, if you do have information on vaccination status, you can do things like stratify and try to look at the attack rate in unvaccinated individuals and whatnot. I think the other thing is, if you're using an assay that has, I think it depends a lot on the question that you want to ask, but if you want to look at things like recent infection, then actually an assay with a lot of weighting could be quite useful, right? It's not going to tell you about seroprevalence, but maybe it can tell you something about recent infection. So I think, yeah, it depends kind of on the question that you're trying to answer.